channel. If this is your first time on this channel, kindly subscribe and press the notification bell beside the subscribe button to get notified anytime a new video is uploaded. Thank you.
I welcome you all to today's lesson. The topic before us is classification of living organisms. Classification of living things. What is classification? Classification in biology is the grouping or sorting or arrangement of living things into the group they fit in best as a result of evolutionary trend, the similarities or the differences that the living things show. In the early age, all living things were put into two groups as animals and plants. There are hierarchy of classification, that is the hierarchical arrangement or hierarchical order of classification of living things. And the largest classification of living things is the kingdom. After kingdom, we have phylum. If it is in animal classification, and if it is in plants, we have division. Thereafter, we have class, followed by order, we have family, we have genus, and finally, we have species. This is what I mean. So this is what I mean. We have kingdom, the largest classification in the hierarchical order. We have the phylum in animals, we have division in plants, we have class, we have order, family, genus, and the species. Species is the lowest or the smallest division in hierarchy of classification. Calvin Linne puts all living things into two kingdoms as kingdom Plantae and kingdom Animalia. But in 1969, there was another man who came up. His name is R.H. Whitaker. R.H. Whitaker. He was the one who proposed 5 kingdom classification that we have till today. All right, this is the 5 kingdom classification that we have presently, and it was proposed by the man R.H. Whitaker. And these 5 kingdoms are from the smallest to the most advanced. We have kingdom Monera, we have kingdom Protista, we have kingdom Fungi, Plantae, and finally we have kingdom Animalia. This is an advancement over what Calvin Linne did, which was 2 kingdom classification as Plantae and kingdom Animalia. Now, what are the reasons for classification? Why is classification so important? Number one reason that classification is important is to easily identify and make references to each living organism. Number 2 is to draw a scheme that shows the differences and similarities between different living organisms. Number 3 reason is to trace the possible origin of individual organisms. And finally, for the purpose of research. The next thing we're going to be talking about is binomial nomenclature. Bi means 2. Binomial nomenclature simply tells us, as proposed by Calvin Linne, that every living organism must have 2 names. This man initiated this with himself by Latinizing his own name. He Latinized his name from Calvin Linne to Carolus Linus. And in the process, he made the following proposals: that every living organism must have 2 names. The first name is the generic name. The second name is the specific name or specific epithet. And the first name, which is the generic name, must begin with a capital letter, while the specific name must begin with a small letter. Not only that, when it is handwritten, the 2 names must be written separately and must be underlined separately. And the next one is that when it is written in italics, the 2 names must be written separately, with the generic name starting with a capital letter, the specific epithet starting with a small letter, but must not be underlined. And this rules valid till today. And that is why in the zoological world and in the botanical world, we have every living organism having two names. You either call it botanical name or you call it scientific name. Let's take, for example, the common rat in the house is Rattus rattus. This is what we are talking about. This is the generic name. It begins with a capital letter. Rattus rattus. And that is rat. We have Musca domestica, that is the housefly.
Finally,
we have Adansonia delimitata,
and that is two kingdom classification as Plantae and kingdom Animalia. Now, what are the reasons for classification?
Why is classification so important?
Number one reason that classification is important is to easily identify and make references to each living organism. Number two,
our spear grass, and so many others. Please don't forget that presently we have 5 kingdom classification: Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, Kingdom Animalia. And as proposed by Carbonline, who Latinized his own name from Carbonline to Carolus Linus, we have binomial nomenclature, which means that every living organism must have 2 names. The 2 names must be written separately and underlined separately, with the first name beginning with a capital letter, the second one beginning with a small letter, because the first one is the generic name and the second one is the specific epithet or specific name. When it is written in italics, the 2 names must be written separately, with the first name beginning with a capital letter and the second one, specific name, starting with a small letter, but must not be underlined. And this is what we have on the board. Now we move on to the next phase. Let's have example of what this classification is properly. Let's classify man as an example of what we are saying. This is a classification of man. Man belongs to kingdom Animalia, phylum Chordata, class Mammalia, other primates, family Hominidae, we have genus Homo, and species is Sapiens. And that is why it is very common to say the scientific name of man is Homo sapiens. Now, there is one thing that we need to understand very well. At times, there could be prefixes in some of these group of classification, in some of the hierarchies of classification. For example, we could have subphylum, we could have superclass, we could have subclass. Let's take, for example, man belongs to kingdom Animalia, phylum Chordata, subphylum Vertebrata, meaning that it has backbone. Man has backbone. Don't forget hierarchy of classification. We have kingdom, we have phylum, division in plants, phylum in animals. We have class, we have order, we have family, we have genus, and we have species. And at times, we could have some prefixes added to some of these hierarchies, such as superclass, subclass, subphylum. Now, we proceed by taking the kingdom one after the other to explain what they are, what they mean, and what is in the intention of studying these organisms. The intention, the reason why we need to understand them. Number 1 kingdom we're going to consider is kingdom Monera. Kingdom Monera contains organisms that are the most primitive. And that tells us that kingdom Monera is the most primitive among the kingdoms that we have, meaning that it contains organisms that are very, very simple. In this kingdom Monera, there are 2 divisions. Division number 1, we call it Schistophyta. Number 2, we have Cyanobacteria. We have Schistophyta and Cyanobacteria. The Schistophyta consists of organisms that generally called bacteria, while the cyanobacteria consists of organisms that are generally called blue-green algae. The representative of Monera is the bacteria. So we're going to major this class, we're going to major this teaching on bacteria. Number one, let's look at the characteristics of bacteria, which are also the characteristics of the kingdom Monera, because we are considering bacteria as the representative of that kingdom. Number one, they are prokaryotic organisms, or you simply call them prokaryotes. They are prokaryotes, or they are prokaryotic organisms. There are two spellings to this prokaryotic, and the two of them are correct. Look at the spelling, K-A-R and C-A-R. Prokaryotic or prokaryote simply means organisms with no true nucleus. That can be said in another way as organisms that do not have membrane-bound nucleus. Normally, when you draw a cell in biology, you draw it this way. You say this is the cell, that's the cell membrane. Then you have this is the nucleus. And this is the nuclear membrane. This is a true nucleus. We call it eukaryotic cell. But for bacteria in kingdom Monera, they do not have this membrane. They do not have nuclear membrane. And that is why we call them prokaryotic cells. This is an example of a bacterium cell. This is supposed to be the nucleus, but there is no membrane around this nuclear material. Definitely, it is a prokaryotic cell. What is a prokaryotic cell? A prokaryotic cell is a cell that does not have true nucleus. That is correct. And it can otherwise be said that it is a cell that does not have membrane-bound nucleus. I hope you get that. All right, let's move on. Number one characteristic of bacteria is that they are prokaryotic cells. Number two, they are unicellular, meaning that it is only one cell like this. This is a cell. Number three, they are microscopic. You can't see them with the ordinary eye. They can only be seen through the microscope. Number next, they reproduce asexually by binary fission. They reproduce asexually by binary fission, meaning that a cell will divide mitotically into two. Two cells will divide into four, four will divide into eight, eight will divide into 16, 16 into 32, 32 into 64, and so on like that. That is the meaning of binary fission. But don't forget, the principle behind, the cell division behind this binary fission is mitotic cell division. So they reproduce asexually by binary fission. The next thing is that some are beneficial while others are pathogenic, meaning that some are of advantages to other organisms. Maybe the decomposition of materials naturally, in the production of some items in the industry, maybe in the production of yogurt, in the resin of fiber, and so on. They are useful. Even in digestion, they are useful, while others are pathogenic, meaning that they cause diseases. They are harmful to other organisms. The next thing is that we have some that are motile, meaning that they can move. Those ones that they can move, they possess cell organelle known as flagellum. Flagellum is a locomotory organelle and not organ, because this is a unicellular organism. The components of the body of a cell are organelles and not an organ. Before you can have an organ, you must have many tissues that are working together to perform a similar function, a specific function. So there are no tissues here. It is as simple as a cell. So the body components are known as organelle. I want to believe that you are here with me. Anytime you see a structure like this in biology, it simply represents a bacterium, a unicellular, microscopic, prokaryotic organism or a cell that may be motile or non-motile, and it reproduces asexually by binary fission. Some of them are beneficial while others are pathogenic. Types of bacteria based on shapes. Number one, bacteria can appear as a sphere in spherical shape. When it is only one like this, we call it coccus. This is singular form. The plural form is cocci. When sphere occurs in pairs, we call it diplococcus or diplococci. These spherical bacteria can occur in chains. When they occur in chains, we call it streptococcus or streptococci. When this spherical bacteria occurs in clusters, when they appear in colony, we call it staphylococcus. And then we have another group. This is the spherical group in spheres. Number two, we have some that are in comma shape. They look like comma. When you see a shape like this, it represents a type of bacteria called Vibrio. That's Vibrio. Number three shape, we have the rod-like shape. When it appears this way, we call it bacillus or bacilli. And just like we have in spheres, when it appears in chains, that is streptobacillus. That's streptobacillus. It is possible for this rod-like shaped bacillus to also occur in colony. When they occur in colony or when they occur in cluster, we call it Staphylobacillus. I hope you understand that. Now we move on to number four, we have the spiral shape. When it is spiral shape, we're talking about something like this. We call it spirillum. That's the spring form. Don't forget that bacteria can exist in different shapes. Number one, we have the spherical or the spheres, and it could be coccus, diplococcus, streptococcus, or staphylococcus. Then we have the comma shape, the vibrio. We have the rod-like, which is the bacillus, and we have the spiral form, which is the spirillum. We still have other group of bacteria. Based on utilization of oxygen, we have three types of bacteria. Number one, we have aerobic bacteria. Number two, we have anaerobic bacteria. And lastly, we have facultative bacteria. Aerobic bacteria are the bacteria that can only live or survive when there is oxygen or in the presence of oxygen. An example of this is Mycobacterium tuberculosis, the causative organism of tuberculosis. Another one is Pseudomonas, that is involved in the nitrogen cycle. Now, for anaerobic bacteria, they are the bacteria that does not require oxygen for their survival, meaning that they survive where there is no oxygen. That is anaerobic bacteria. Example of this is Clostridium. Clostridium that is involved in fixing atmospheric nitrogen non-symbiotically. And lastly, we have facultative bacteria. These are the bacteria that when oxygen is available, they make use of it. And if oxygen is not available, they respire anaerobically. Example of this is E. coli. Now we move on to the next group of bacteria or the next step of bacteria. We are going to the next group of bacteria based on their response to gram stain. The original color of gram stain is purple. When you add gram stain to a known source of bacteria, and when viewed under the microscope, you see the bacteria retaining the purple color, we say that it is gram positive. But when it turns pink, it shows that it is gram negative. All right, based on gram stain, there are two groups of bacteria. We have the gram positive, which takes the original color of the gram stain, that is purple. And then we have gram negative. When the gram stain is added to a known source of bacteria and it is turning pink, it shows that it is a gram negative because it's going to turn pink. This is very, very significant in pharmacology, in pharmacy, in biochemistry, science, and medical laboratories in order to dictate what type of bacteria is this, or what type of bacteria this is, in order to know how quick attention should be given, especially to the pathogenic, and what type of antibiotics to be given. This is very, very useful in determining or detecting or diagnosing bacterial diseases and providing antibiotics or treatment as urgent as possible, depending on the absorption of this gram stain by the cell wall of the bacteria, which is made up of peptidoglycan, the combination of protein and carbohydrates. I want to believe that you really understand what I'm putting across to you. So don't forget that there are 5 kingdoms of living organisms, and we have 7 hierarchies of classification ranging from kingdom to division or phylum. We have class, we have order, we have family, we have genus, and we have species. Don't forget binomial nomenclature, and don't forget that it was Whitaker, R.H. Whitaker, in 1969 who proposed the 5 kingdom classification that we still use today. And before him was Carolinus, who was formerly Cavonline. He Latinized his own name, and he was the one who proposed the 2 kingdom classification that were used earlier before Robert H. Whitaker came up with 5 kingdom classification. And we have started to look into this classification starting from kingdom Monera, the most primitive. And we said that the representative of this group is bacteria. We have seen their characteristics, we have seen this different shape that they have, and we have learned that some can be pathogenic or they can be beneficial. Thank you very much for watching. Thank you very much for listening. Thank you very much for being part of this lesson. I want to tell you that we're going to meet next week for beautiful edition of this biology on this channel, My Biology. Till then, keep loving, keep reading biology.